thought I'd provide a little background about myself and kind of how I arrived here. So, majority of my family is from Ireland, and many of them are still living there. I was not born there, but supposedly I was conceived there, and it may have involved some psilocybin mushrooms and a Bob Dylan concert at a castle, but that's a story for another time. So, but eventually, I made my way to Jersey City, where I was born to my adoptive parents there, and this is the view from the Jersey City Hospital here. And my dad worked for the government, and so we moved around quite a bit, and eventually moved to a little barrier island off the coast of Georgia, southern coast of Georgia, called St. Simon's Island, Georgia. And that's really what kind of sparked my interest in veterinary medicine. It wasn't very developed at the time. There was just a law enforcement training center located nearby, which is how we ended up there. And so I'd spent a lot of time just in the woods collecting animals, snakes, turtles, the whatnot, and really, thought I wanted to be a veterinarian at that time. But again, we moved associated with this job, and this time we landed in Cleveland, Ohio. And it's really not as bad as you think. So it's not all burning rivers and lake effect snow. It is gray for many months of the winter, like today. But so that kind of sparked a secondary interest in mine when I arrived there. So I really hadn't seen snow, I guess. I was really young when I left New Jersey, and I became fascinated. I didn't have any friends either when we moved there. So I'm watching the Weather Channel all hours of the day, and became fascinated with winter weather. And so I thought, that was something I wanted to pursue for a career. And so I actually went, long story, but had some family still down in Georgia, went back down to the University of Georgia, had a good meteorology program. And I thought I'd be a winter weather specialist and perhaps pursue a PhD in winter weather. I felt I wasn't smart enough to be a veterinarian. And so my goal was to be a skiing weatherman. So this could have been me, right? But here I am instead. No. So, eventually, after about 1 year, meeting a bunch of people in the dorms, the guy across the hall was going to be a cardiothoracic surgeon, and he was really living it up on the weekends. I thought, you know what? I think I could be a veterinarian. And so I'm smart enough. So I changed my course and completed my DVM there, as Sue mentioned. And when I finished that up, as I'll talk about, I had this interest in cancer research by then. And so, I was looking for, and pathology as well. And so I was looking for training programs, and I was really drawn to CSU and their reputation, especially of the department, of training, veterinary biomedical scientists through this combined pathology program. And then obviously the Flint Animal Cancer Center, and then the great group of people there and all the work they were doing. And so that's how I came to Fort Collins in 2011. But, as they say, old habits die hard. So I still like to do a lot of skiing, and that might have been involved in part of my reason to attend here. So, but kind of coming back to my research quickly here. So, I was a late bloomer. I was, first generation college student. Nobody had really gone to college. Didn't know that landscape. So I didn't get involved in research until second year of my veterinary training, essentially. It was this opportunity through an ophthalmology class to write a grant proposal, but I'd become really fascinated in immunology, and so I wrote this proposal to study the role of T lymphocytes, these Th17 cells involved in autoimmunity, in this recurrent uveitis syndrome in horses. And so that was my first foray into research, and again, a lot of great mentors there helping me and encouraging me. And so that was a really great experience, but at the time, as I was doing that, my father was diagnosed with cancer. And so I started to kind of dive into the cancer literature as well. And so I realized this huge field of tumor immunology was really kind of blossoming at the time. This was 2008, 2009. And so I loved the autoimmunity work, but I realized I really wanted to focus my work on cancer immunology. And it was really this paper here that I became aware of at this time. It came out in 2005, this Nature paper, but I didn't really come aware of it till 2009, about this idea of what's called a pre-metastatic niche. And so this is this concept where a primary tumor can kind of remotely deploy immune cells from the bone marrow to colonize these distant future metastatic sites, let's say the lung, ever before the arrival of a single tumor cell. And so, I came across this. My father's battling cancer. I'm working in immunology, and then I see this concept, and it just blew my mind. And I thought, I have to work on this. And so I arrived at CSU with this crazy preconceived notion that I need to work on this concept. And that should be a red flag for anybody recruiting graduate students right there, but not for Steve Dow. So Steve's like, yeah, sure, join the group, let's do it. And he was, I'll tell you, working on it, and I'll get to that in a moment. So maybe the only person crazy enough to take me into his lab. But anyway, so this 2005 paper, though, I really want to spend some time on this, was kind of the first paper to provide some molecular evidence of this concept called the seed and soil hypothesis. So this was a hypothesis originally postulated by the British surgeon Stephen Paget in the late 1800s. And so he had reviewed maybe 900 cases of breast cancer, just patient medical records, necropsy records. And as he's going through and looking at cause of death, which is obviously from metastasis, he noticed that these metastases were at very specific sites, specific organs. So he would find them in the lung or the brain or the bones sometimes. You wouldn't see them in other also highly vascular organs, right? You would never find patients dying from metastases to the spleen or the kidney or the adrenal glands or anything like that. And so he was the first to kind of postulate this idea of this organ site specific metastatic spread, right? So this concept that it wasn't just something about the seed or the tumor cells themselves that dictated where they spread in the body and killed the patient, but it had to be something about those recipient organs and the soil of those organs, right? The cell types that were in those organs that made it a hospitable environment for these tumor cells. And so, again, learning about this kind of seed and soil hypothesis and this mechanistic paper and being a veterinarian and training it, immediately piqued my interest because I was learning about this cancer called osteosarcoma, the specific type of bone cancer. And you're taught, right, immediately in veterinary school that this tumor has this very strong predilection for spread to the lungs, right? So these dogs will present with this cancer. It's they'll have clean radiographs of their lungs. It's primary localized tumor to the bone, but you're told that 90% of these dogs have micrometastatic disease in the lungs and this strong predilection. So that really just piqued my interest. And so just to kind of provide some background, this is really what we focus on, but it's the most common primary malignant tumor of bone. It's primarily a pediatric disease, and even though it is the most common malignant bone tumor, it's still a very rare cancer. So we only see about 800 cases of this a year in the US in people. But the major clinical problem with this disease is that, while 60 to 70% of patients do really well with our current standard of care, 30 to 40% of these patients will fail that therapy. And they'll fail it very rapidly and almost exclusively in the form of lung metastases. And so once that occurs, and once these patients have relapsed with these lung metastases, their survival time is significantly diminished. So roughly 20% of these patients will make it 5 more years. So this recurrence happens about 1.5 years, and it's deadly. And we have no therapies to offer these children. So, and that hasn't changed for 40 years. Okay, so that's really the major problem here and what we're focused on. So, initially I was pretty naive to think I was the only veterinarian that had made this connection between bone cancer in dogs and metastasis. But as I dove into the literature, became aware of this large comparative oncology community and all this great work coming out of places like CSU and others around the country that were doing all the work to kind of build the dog as a model for osteosarcoma. So I just want to kind of touch on that. So these groups were demonstrating that these tumors were very, very similar on the histological level, on their clinical presentation and progression, and even genomically, right? Driven by the same driver mutations. Or here, if you're looking at this heat map, if you did whole transcriptome sequencing of these canine and human tumors, these tumors would intermingle or cluster together, right? They would not cluster separately. And so this is a lot of great work already done by the field to really establish dogs with this bone cancer as perhaps the greatest single model of a human solid tumor counterpart. And so this foundation is really the focus of our laboratory, right? This idea of the seed and soil hypothesis and these tumor microenvironments and the cell types of that soil promoting metastasis, and specifically within the context of osteosarcoma. And so that's what I'm going to talk about, and what our overall goal is, to kind of take this multi-pronged approach to understanding and trying to target these mechanisms to slow or prevent osteosarcoma metastasis. So the first story I'm going to talk about is this population of chemokine receptor 2 or CCR2 expressing monocytes. So again, when I came here, I joined Steve's lab, and he had already just recently finished conducting this in silico screen, looking for small molecule drugs that were off patent, FDA approved, so readily available and cheap, that could be repurposed as monocyte migration inhibitors through antagonizing this CCR2 receptor. And so that was really when I had joined the group, and Steve at the time had identified one such compound called Losartan, which 50 million Americans take to monitor or to regulate their blood pressures. This is an antihypertensive drug, and so that was my kind of role in the lab, was to jump in and start screening this drug to see if it could inhibit monocyte migration. And so another one of the first projects in the lab was to also start surveying lung metastases from common and highly metastatic canine tumor types to see if we could understand which of these tumors were reliant on these monocytes and this CCL2, CCR2 mediated recruitment. And so one of the tumors that we observed to be very enriched in CCL2 expression and infiltrated with these monocytes was osteosarcoma. And this was something we observed both in canine osteosarcoma shown here. These are monocytes in red, and this is cytoplasmic CCL2 expression in red in tumor cells. But we also observed this in human osteosarcoma tissues and cell lines, which would overexpress this monocyte chemokine CCL2. And when they did, they were very efficient at recruiting this population of monocytes. And so, like I said, I'm working with this in silico identified compound Losartan, and we're screening it in vitro monocyte migration assays, and then we're also leveraging mouse models, and these were CCR2 knockout mouse models, or mouse models that were knocked out for Losartan's intended antihypertensive receptor. And so, we would observe that if you gave mice early lung metastases, or if you vaccinated this mice, you would get this strong influx of these monocytes into tissues. And then if we treated these mice with either Losartan or we knocked out the receptor, we could inhibit this process. And again, this is data just showing that, flow cytometry data. These are these CCR2 monocytes of interest here. And again, Losartan treatment could reduce these levels to near genetic knockout of this receptor. And again, when you would inject tumor cells into the lungs of these mice here labeled in green, you would get this influx of monocytes. And again, Losartan could inhibit that process, which is quantified here. And so similarly, we, obviously being the primary goal, we moved Losartan into these more longer term mouse studies looking at lung metastasis progression over time. And so again, we could treat these mice daily with Losartan and block these monocytes and get this sustained monocyte blockade, and it would significantly slow down metastatic growth. And that's what we're showing here. So again, this wasn't a silver bullet. We weren't stopping metastasis formation, but we were definitely slowing down its growth, which you can see quantified here, and histologically here, these are these purple lung metastases. And so at this point, we were pretty confident in this preclinical data that we had identified a repurposed therapy that could block this monocyte recruitment. And when you did this, you could slow lung metastasis development. And this was observed in mouse models of breast cancer, colon cancer, and osteosarcoma as well. But again, we knew it wasn't a silver bullet, and we weren't able to completely prevent this. And so we wanted to combine this with another therapy, which we chose was Tesseraneb. So at the time, Tesseraneb was the only veterinary approved tyrosine kinase inhibitor and anti-cancer drug in veterinary medicine. And there was a lot of work around this Tesseraneb drug and how it had effects on this molecule called STAT3, which was really important for myeloid cell generation and differentiation. And a lot of other literature, a lot of other studies in the literature had shown that Tesseraneb, in addition to its known inhibitory effects on these tyrosine kinases, could inhibit STAT3 and block these myeloid cells. And so this was a real rational combination for us to combine with Losartan. So obviously we moved this therapy into a clinical trial now in dogs with lung metastatic osteosarcoma and run right here at the Flint Animal Cancer Center. And so to really kind of just summarize this and long story short, we started this trial at the recommended dose of Losartan in dogs, which was used again to treat high blood pressure. And this was a pretty low dose of 1 mg per kg. And when we did that, we really didn't see any responses like we were seeing in these mice, right? So we saw some stable disease that's denoted here, but not the type of effect we were seeing in the mouse models. And so again, through doing some PK, measuring drug levels in these dogs, we were able to modify this trial and increase the dose tenfold to finally get this drug at levels we were using in our mouse models. And it was still very safe and well tolerated in these dogs. And when we did, we started to actually see some clinical benefit in these patients. So 25% of these patients were getting actually regression of their metastases. And so really at this point, right, this is a late stage of this cancer, this metastatic disease. If you do nothing in these patients, they're dying in 30 to 40 days. So typically nothing is really effective at this point in disease progression. And so when we were seeing 25% of these lesions reducing in size and another 25% of dogs getting stable disease, we were really excited about this. And eventually presented this data at the Children's Hospital in Colorado. And when they saw this data, again, they likewise were also very excited. And so ultimately, this canine data has led to direct translation of this drug combination to a human metastatic osteosarcoma trial. So Tesiranib, like I told you, it's a veterinary tyrosine kinase inhibitor, but there's a drug approved for human osteosarcoma called sunitinib, and they're functionally equivalent, and so we were able to kind of quickly translate this drug combination. So this is ongoing right now with Children's Hospital of Colorado and Children's Healthcare of Atlanta, and we're kind of in the middle of this dose level 2 here in terms of patient enrollment. But what's been encouraging for us is that, again, we can go in and we can use flow cytometry here, and we can look at this population of CCR2 expressing monocytes, and we can see with this therapy that we're able to significantly reduce this same target, right? The same pharmacodynamic endpoint that we were modulating in the dogs, we're able to inhibit in these human osteosarcoma patients. So, okay, so now I kind of want to shift gears and talk about another cell population within this lung metastatic tumor microenvironment, and this is this population of lung fibroblasts or activated lung fibroblasts. And so this has really been work primarily driven by Eric Palmer, who's an MIT PhD student in the lab over the past approximately 4 years. So Eric's work is primarily focusing on these vesicles called exosomes or extracellular vesicles. So exosomes are these small nano-sized, approximately 100 nanometer phospholipid membrane-bound particles. They're released by all cells of the body, but they're also released by tumor cells, and they're really important or critical mediators of intercellular communication. So these phospholipid-bound particles are packed with biological cargo, nucleic acids, proteins. They can even carry whole mitochondria, and they can function similar to hormones. So they can travel long distances through the blood, throughout the body, and mediate intercellular communication, and specifically remotely prime these distant future metastatic sites and start to change the phenotype of cells at that site to create a permissive metastatic environment. It's just a video of these from our laboratory here. So Eric's really done over these last few years a phenomenal job kind of bringing this expertise in the lab. So this wasn't something I had worked on for a PhD, but knew if we wanted to study this phenomenon, we had to bring this into the lab. And so he's optimized effective isolation of a pure population of these vesicles, so you can size them here, and we see that, yes, indeed, they have that prototypical size around 100 nanometers. They're expressing some of these classical membrane exosome markers, which are denoted here by flow cytometry and Western blot, and they have the classic morphology by EM evaluation. And so one of the first cell types, again, once we were able to isolate these, that we wanted to look at, how are these priming and affecting cell types of the lung and of the metastatic lung was lung fibroblasts. And so, again, we observed in vitro that Eric could isolate exosomes from a panel of 6 different human osteosarcoma cell lines and work across 3 different primary, these are primary human donor-derived lung fibroblasts, both male and female. and we'd see robust uptake of these particles into the cytoplasm of these cells, both by microscopy here and flow cytometry here. And again, our primary interest was in the immunological response of these cell types, right? And so really, to look at this, and don't have time to get into it, but a lot of these hypotheses were based on drawing parallels with another lung disease called idiopathic pulmonary fibrosis. But to look at how these fibroblasts were responding, we first took a pretty simple discovery approach, just running a multiplex cytokine assay, looking at how these exosomes affect cytokine secretion by these fibroblasts in a panel of 48 different analytes. And so surprisingly, when we did this, 3 major molecules popped up, which are summarized in this table. But this was, again, CCL2, the Losartan target, IL-8 or CXCL8, and then IL-6. And interestingly, obviously we had demonstrated a role for CCL2 in osteosarcoma metastasis, but these other 2 molecules had also been recently published as major mediators of osteosarcoma metastasis by another group at Nationwide Children's Hospital at Ohio State. And another thing that was also interesting to us is that once we observed this, we were curious, is this an osteosarcoma cell exosome-specific phenomenon or just something that's conserved across cancer types? And so we went and screened a number of other human highly metastatic tumor types, so spanning melanoma, colorectal cancer, bladder cancer, breast cancer. And so you can see for the most part, especially for some of these molecules, this seems to be an osteosarcoma-specific phenomenon here. And so one of our major goals was to kind of understand the molecular mechanisms that were regulating this exosome activation of these fibroblasts and this chemokine secretion, obviously to try and interrupt that therapeutically. And so this is kind of ongoing. We're taking a number of approaches, looking at both membrane-based and bead-based phosphokinase arrays to look at what are some of these signal transduction pathways being activated in these fibroblasts. And so far it's been mixed results, but one of the molecules somebody else in the lab had been studying that we were interested in looking at is this other non-receptor tyrosine kinase called focal adhesion kinase. So focal adhesion kinase, and the reason we were interested in looking at this as a potential target of these exosomes and these fibroblasts is that it serves as a biosensor and integrator of extracellular signals, and specifically of integrin signaling, and integrin receptors are known receptors for exosome uptake in cells. And so indeed, when we looked at this by Western blot, we can see that there was phosphorylation of FAK or focal adhesion kinase in these fibroblasts, albeit this seems to be constitutively activated in these cells and not necessarily induced upon exosome treatment here. And so the reason that FAK still was intriguing to us is some of these multiple pro-tumorigenic functions it has, and specifically in other tumor types, individuals have shown that it could drive chemokine secretion. And still, so supporting this data, we've generated some single-cell RNA sequencing of canine lung metastases, and we've also observed that FAK is significantly upregulated in fibroblasts within these metastases, and that's expanded here, and FAP expression is denoted in these purple hues here. And that, when you look at this at a cell type, on a per cell type basis, fibroblasts are the second highest expressors of focal adhesion kinase within this lung metastatic microenvironment, and these fibroblasts are also the top or primary source of this CCL2. So this canine data in these lung metastases seems to really mirror some of this in vitro data we're observing in our human osteosarcoma studies. And again, to kind of corroborate this, right, this was just a single canine osteosarcoma metastases that we were able to get for single-cell RNA sequencing. We went back into some publicly available bulk RNA-seq data where we could look at 50 canine osteosarcoma tumors and 18 human osteosarcoma tumors. And so when you dichotomize this larger number of tumors based on activation of this focal adhesion kinase pathway, we would see significant enrichment in many processes associated with an immune suppressive microenvironment. So not sure if everybody can read these, but if you can, you're seeing things like IL-8 pop out, monocyte macrophage recruitment pop out, TGF-beta secretion, right, indicators of fibroblast activation. So a lot of parallels suggesting that FAK activation in these fibroblasts could really start to drive this cascade of events that leads to this chemokine secretion, enrichment of these monocyte and macrophage populations in these lung metastases, and this overall immune suppressive tumor microenvironment. And so we really wanted to, again, target this process. And so we found a FAK inhibitor, or focal adhesion kinase inhibitor, called defactinib. And what we could do, again, we went back to our in vitro fibroblast assays where we're educating these cells with osteosarcoma-derived exosomes, which we observed would induce significant proliferation of these fibroblasts, and again, significant CCL2 secretion. And when we treated these cells with defactinib at these pharmacologically achievable concentrations, we could both inhibit this proliferation, which is shown here and quantified here, and block this chemokine secretion with this compound. And again, another reason we think these fibroblasts are important, so not just through their ability to make these chemokines and manipulate this tumor microenvironment, but Eric's done some really nice work showing that they seem to have this direct pro-survival effect on these osteosarcoma tumor cells. So these are growth assays. These are serum-free or serum starvation growth assays where we can seed human osteosarcoma cells on top of these lung fibroblasts, either left naive or primed with these human osteosarcoma exosomes. And so what you see when you monitor this growth over a period of 6 to 7 days is that these exosome-activated lung fibroblasts provide a significant growth advantage to these osteosarcoma cells under this condition of serum starvation stress. And that's quantified here. Again, that's conserved across 3 different cell lines and 3 different lung fibroblast donors. And there's images here depicting this phenomenon here with these tumor cells labeled in red. And so importantly, this FAK inhibitor also seems to have a direct anti-cancer effect on these tumor cells. So we've observed constitutive FAK activation in osteosarcoma cells themselves. I'm sure you saw that on some of the UMAP plots of the single-cell RNA-seq data. And again, we can treat these cells with reasonable concentrations of defactinib and get significant direct cytotoxicity. And again, if we move this into one of our mouse osteosarcoma metastasis models, even just single-agent defactinib can significantly prolong survival of these mice. And so this data now has kind of led to the second generation of this Losartan tasitinib therapy, which is an addition of this defactinib compound for dual targeting of lung fibroblasts and macrophages in metastatic osteosarcoma. And so to do this so far, we've conducted pharmacokinetic studies of defactinib in dogs. So this compound's in phase 1-2 trials in people. It's never been in dogs besides tox studies. And we've observed really favorable PK, so the drug seems to be well tolerated. We can achieve very, very high levels of the drug by administering it orally and exceed those levels we know to be required, at least in vitro or in our mouse models, to kill these tumor cells or modulate chemokine secretion. And so this trial is now open in dogs with osteosarcoma. We've so far just enrolled 1 patient, opened a few weeks ago. So real quick, I just want to touch on this. So, we're looking at defactinib as a mechanism to target these fibroblasts, but in a parallel approach, Marika Kozowski in the lab is going to be looking at chimeric antigen receptor T cells that target a cell surface protein that's expressed by this population of fibroblasts called fibroblast activation protein. And so we've been working with Mike Liebowitz's lab at the University of Colorado, and we've been able to generate canine CAR T cells targeting FAP. And these cells seem to do an excellent job at killing FAP-expressing canine osteosarcoma cells and will express high levels of interferon gamma when they do that. All right, so just finish up here on the last little bit of this talk and the third population of cells that we're targeting, which is the resident alveolar macrophage, right? So I've told you that this population of recruited monocytes seems to play a role in metastasis. We're interested if these resident macrophages can also serve as a reservoir for tumor macrophages, and specifically if they might be an indicator of those patients likely to respond to some of these therapies. So this is work really being driven by a postdoc in the lab, Katie Kronice, and then a DVM PhD student in the lab, Laurel Haynes. So Laurel's done some really nice work in our mouse models showing that, again, these osteosarcoma exosomes will hone to the lungs and specifically seem to have a tropism for uptake in the lungs when you look at them relative to some of the other organs in the body. This is both through IVUS imaging here and then quantifying this process through fluorescent labeling of these exosomes and through flow cytometry. And importantly, Laurel's observed that these exosomes seem to be taken up by a population of alveolar macrophages in the lung. So these are airway accessible macrophages that seem to be taking up these exosomes. And when they do, again, we see upregulation of our chemokine CCL2, and specifically within this bronchoalveolar lavage fluid and not necessarily out in the periphery here. And so some of this early foundational work in these mouse models has led us to this overarching hypothesis, which is, can we leverage a relatively long-standing diagnostic technique, right, of bronchoalveolar lavage? People are normally using this to diagnose pneumonias as an improved monitor or immune correlate of response to these immunotherapies in dogs with osteosarcoma as compared to monitoring these patients in the peripheral blood here. And so this is work that Katie's doing. So we have an ongoing trial as well in dogs where we're looking at healthy dogs with primary localized or pre-metastatic osteosarcoma, and then those dogs with gross metastatic osteosarcoma, and we're performing bronchoalveolar lavage on these patients and performing single-cell RNA sequencing on those cells that we're obtaining from that lavage fluid. And so this is just early data from 3 healthy dogs and 5 dogs with primary localized osteosarcoma. It's 32,000 cells, but Katie's identified 40 distinct 40 unique cell clusters, including 16 different populations of macrophages in this fluid. And when you compare these 3 or 5 dogs with primary localized osteosarcoma to the 3 healthy dogs, 11 of these 16 macrophage populations appear to be upregulated in these tumor-bearing dogs, right? An increased frequency of these tumor-bearing dogs. And again, when we look back at levels of CCL2 in this bronchoalveolar lavage fluid correlate with the total percentage of these macrophage populations in this cell fluid. They do not in the plasma. And again, potentially FAC expression within cell types in this fluid seems to correlate with monocyte abundance. And this is, again, just some early data, but something where we're really hoping to kind of use this as an improved surrogate for immune monitoring and perhaps as a predictor of those patients most likely to respond to these microenvironment targeted immunotherapies. So really just to kind of summarize our goals, right, in our work. So we're looking at these oncogenic regulators that are mediating the composition of this osteosarcoma microenvironment and really that how these cells extrinsically promote metastasis. We're looking to take a combinatorial approach to target that, and typically with drugs that we can move quickly into the clinic and leveraging dogs and then hopefully to humans. And then we're really trying to leverage some of these newer technologies of single-cell RNA sequencing and this innovative strategy of sampling this lung fluid as a surrogate for that metastatic environment that you otherwise can't get to, and see if that can really improve our ability to predict those patients most likely to respond. So with that, ton of thank yous here. Don't have time to get to them all, but the department, Jeff Willis for the nomination for the award. Greg, Ed, Sue over the years. Steve, can't thank him enough. Don't need to get in there, but still work with his group quite a bit. This is the group at Children's Hospital of Colorado running the trial. As Sue mentioned, the DVM PhD program. FACC clinical trials team's phenomenal group of people to work with at the hospital. The list goes on, but 2 things real quick too. This is kind of half of my job, I feel like, in research, and the other thing is leveraging my pathology training and trying to collaborate with people. And I wasn't able to touch on that, but these 2 groups have been phenomenal collaborators to work with. So Jeremiah Easley in the PSRL, and then everybody in the OBRL, and it's been really rewarding to work on all their animal models of disease, orthopedic disease, and again, very heavy and impactful translational research. And so couldn't get into that, but definitely wanted to touch on that as well. So thanks again, everybody, for the time, and really appreciate the opportunity to speak about our work.